yesterday we had established two things. One, bacteria do in fact perform protein synthesis. They have to. They have proteins. They have ribosomes. They got to make them somewhere. And we had kind of reviewed the process of protein synthesis in eukaryotes. What are eukaryotes? You! And this plant. And an earthworm. And a smallmouth bass. And a wood duck. And a firefly. And a butterfly. And a dandelion. And a goldfish. And a mouse. And a pinworm. Ooh, we'll talk about those later. Those are all eukaryotes. The only thing that and a mushroom is a eukaryote. The only thing that isn't a eukaryote is a bacteria. Yeah, the prokaryotes are like your bacteria. So we briefly reviewed this process of protein synthesis. DNA unzips. We make an mRNA copy of a section of the DNA. It leaves the nucleus, goes out to the, where do we make proteins? Ribosome. Question? No? You yell if you've got a question, please, please. Goes out to the ribosome, tRNA, the little trucks, the truck RNA. It's actually transport RNA, but I think it's funnier if we say truck RNA. Starts bringing amino acids over to the factory. The ribosome actually does the sort of like crocheting them together. This is all good. Makes a polypeptide chain, gets to a stop codon. What happens? It stops. That couldn't be much more self-explanatory. Get to a start codon, what do you do? Start! Get to a stop codon, what do you do? Stop! This is easy. How many of you have taken a driving test yet? Okay. Green light means go. Red light means stop. Yes, sir.
Yellow light means for.
So, we're asking this question. Thinking back to that process of protein synthesis in eukaryotes, which step can't happen in a prokaryote? Don't yell it out. Circle the step that cannot happen in a prokaryote. You should have these in your notes. And then compare it with your neighbor. Who says it is not possible for bacterial DNA to unzip? Any votes for that one? Good. That's totally possible. You got DNA, you can unzip it. That's easy. Who says it's not possible for bacteria to copy DNA? Or, I'm sorry, for bacterial mRNA to make a copy of DNA? Yeah, you're right, that's totally possible. Who says it's not possible for mRNA to leave the nucleus in a bacteria? Yeah! Why? Oh. Well, that's a minor complication. Yeah, bacteria don't have a nucleus. So we don't get that step in bacterial DNA synthesis. That's really your first big difference. So is this a problem, or does this actually make this process a little bit easier and more efficient? It actually makes it more efficient. Now, bacteria still make an mRNA copy, because they don't have to copy that whole loop of DNA. They still only need a little section of DNA. You don't want to copy the whole thing, you just want the piece that you want right now. This is like if you've got a giant cookbook and you're just making banana muffins, you don't need to copy the whole book. You can just copy one page and take it to the kitchen with you. Okay? Okay, this is a lot of text, so I'm going to read it and then shut up and let you copy. This is a comparison of protein synthesis in prokaryotes and eukaryotes. These are things that only happen in eukaryotes. Plants, mushrooms. These are things that only happen in bacteria, prokaryotes. And these are things that actually happen in both of them. These are the similarities. So this is sort of a compare and contrast. Do you want me to let you copy and then I'll read it? I'll do that. Okay, so what you've got coming around is it's a compare and contrast organizer. Anybody know the real name for a diagram like this? Venn diagram. Good. And we'll kind of work on this one together. They're great. Venn diagrams are great for comparing and contrasting because you can look at things that only happen in one case as well as things that happen in both situations. And that's exactly what we're going to do. So I'm just going to discuss this real briefly, and then we'll do the Venn diagram together. So you notice that in both eukaryotes and prokaryotes, we use mRNA. Every living thing on the planet that we know of, we've never found a living thing that doesn't use RNA in building proteins. And every living thing on the planet builds proteins. Even bacteria, the simplest little tiny things. Every living thing we know of uses RNA and builds proteins. That happens in everything. Here's the first big difference. In a eukaryote, that mRNA has to leave the nucleus. Because the DNA is too bulky, too big, it's got too much information on it to come out of the nucleus. It's neatly stored. We don't move it if we don't have to. There is, so protein construction doesn't happen until the mRNA gets out. In bacteria, there's no nucleus. There's no door to get through. So the minute that you have completed codons on one end, it starts making protein. Boom, boom, boom. It's much faster this way. You don't have to wait for anything. You're not waiting on something to go on. So protein construction starts immediately. They actually call this transcription translation coupling. Because what's the step what is transcription? Going from what to what? DNA to mRNA. Translation is going from the mRNA to proteins through the use of tRNA. And in eukaryotes, they're very distinct steps. They happen in different places. There's a waiting period. In bacteria, it's happening at the same time. On one end, it's making mRNA. On one end, it's starting to make proteins. On the other end, it's still building the mRNA. So it's like reading the book while somebody's still writing it. It's kind of cool. But in both cases, we're making mRNA from DNA. In bacteria, the mRNA strand is just for one protein at a time. So you remember the video with the dinosaurs and the proteins, and it shows all little amino acids linking together, and they all fold up in you and a maple tree and a goldfish and an earthworm and a firefly and a honeybee and a dandelion. One little protein gets made at a time. In bacteria, that strand actually codes for a couple proteins at a time, which seems to me like it's more advanced and that should be our thing, but it's not. Bacteria do that, we don't. They can make multiple proteins off the same strand all at once. Now in both cases, it's the mRNA that carries codons that tell the cell which amino acids to put together. Here's where, we're more complicated. In your cells, there are 10 different chemicals that are required to start building proteins. In a bacteria, it's just 2. That's kind of cool. What do you think? So, anybody here? Let's see. No, never mind. That's not a good analogy. But in each case, no matter how many chemicals it takes to get started, once the protein synthesis is started, it's the same exact start codon that says start building a protein. It's AUG, school starts in August. In eukaryotes, mRNA has to travel. In prokaryotes, it travels to the ribosome and it's just fed through the ribosome like a cash register tape. In eukaryotes, a chunk of the ribosome actually pops off and comes over to the mRNA. This is like mobile pet grooming. This is the difference between loading your 90-pound overweight elderly lab in the car and taking her someplace to have her nails trimmed and having somebody in a van pull up in your driveway and trim her nails for you. One is a lot easier. So in bacteria, the ribosome actually goes to the mRNA. Because remember, the mRNA is still attached to the what? It's still attached to the DNA. So it can't go anyplace. It can't go out to a ribosome, so the ribosome comes over and attaches to it. And, you know, these are just general, you'd expect this. In eukaryotes, the ribosome's bigger, it's more complicated. In bacteria, it's smaller, it's simpler, just like everything we've said about bacteria, smaller and simpler. But in both cases, ribosomes are still responsible for building proteins. Now, let's do this one together. So we've got a Venn diagram for eukaryotes, prokaryotes, and what happens in both of them. So, somebody give me something that happens in protein synthesis in both eukaryotes and prokaryotes. Anything. True for both. Go ahead.
They both make protein.
Okay, both make protein. What else is true for both of them? Cameron. Both have DNA. Okay, one more for both, and then we need to start breaking out. Both make RNA. Okay. Now, what's something that's only true of one of these? What's something that's a difference? Okay, eukaryote has a nucleus. Is there a logical counterpart to that we would write for the prokaryote? No nucleus. Nucleus? No nucleus. No nukes. Oh my gosh. That's hilarious. Okay, what else? No nucleus. Has a nucleus? No nucleus. What else? What's something that's only true for either a prokaryote or a eukaryote? You know what? I'm going to start doing. Okay, Tyler, have you contributed anything on this yet? Oh, okay. Then I'm going to have you hold. Garrett, did you contribute anything yet? Okay, go for it. Ah, okay. So in the eukaryote, the ribosome. Okay, let's make sure we phrase this well. In a eukaryote, 10 different chemicals are required to do what? Okay. Needs 10 chemicals to start protein synthesis. Is there a corollary? Is there a natural counterpoint that we could write for prokaryotes? Somebody I have not heard from yet on this. And I'm going to start doing shirt color lottery. Prokaryotes only need 2 chemicals to start the process off. Need 2, and we call them initiation factors, or IFs. And hopefully you can write better than I can, because this is pretty terrible. So we need 2 chemicals to start the process off. Okay, so in prokaryotes, we start building the protein the minute there's a completed codon. Start building. Good heavens, let me try this. Okay, so as soon as there are codons, prokaryotes start building proteins. What's the logical sort of counterpoint to that in eukaryotes? What's different in eukaryotes? Okay, let's see. We'll go with gray. I know there was lots of gray. So anybody with gray shirt, who I haven't heard from yet. So we've got Madison, Chase, Peyton, Cameron. Am I missing anybody in gray? Gray, who I haven't heard from yet? 4 of you. Enough to spread it out. I did. I'm sorry. Okay. So, Peyton, Cameron, Madison. Any of you? If you're near another gray-shirted person, you can coordinate your efforts. What's the logical sort of counterpoint? What's different about this process in eukaryotes? So in prokaryotes, they start building immediately, and I think I spelled immediately wrong. I apologize. No spell check on doceri. Didn't you already contribute?
Yeah.
Did I screw something else up?
No, I just know it.
Oh, okay. Do you want to be phone a friend to one of the people in gray shirts? Three people in gray shirts. No pressure on any of you. It's all spread out. If you have a gray shirt on and you don't know it, you can phone a friend. Any one of you can take this. Can bacteria start building immediately? They have to wait for something? Should I switch shirt colors? Gray shirts are not talking to me. Sounds like a team or a side in a battle, like the red coats, the gray shirts. The red coats are coming, the red coats are coming. Oh, good heavens, we're still recording. That's ridiculous. In a eukaryote, protein construction can't start until the mRNA gets out of the nucleus. So protein building has to wait until the mRNA reaches a ribosome. Okay, here's your assignment. You come up with one more for each of these 3 categories on your own. Now, you can coordinate with other students, but whatever you write has to be your own words. So you're coming up with one in each of these 3 categories on your own. They are due on Monday. If you get it done before you need to leave here, you can put it in the box and be off the hook and go enjoy your weekend. It's not a real burden of homework, I don't think. But that's your homework, is to think of one more in each of those 3 categories. That's where we'll stop.